So let's discuss in this next part and answer key questions. And the first one is really to try to understand what's the basis for therapeutic ultrasound-induced immune sensitization. And I took this cartoon from a paper discussing ablation from cryo, microwave, RF ablation. But the idea will be the same with therapeutic ultrasound intervention. And one of the messages is try to convey that the way this intervention will induce tumor cell death is very important to understand what kind of immune response might be triggered. In particular, what kind of DAMPs or danger signal will be released. Will there be co-stimulatory signaling present? And there is a list of questions that we need to answer in order to really understand what's going on. And if you think first about this T cell, we talked about CD8 positive T cells, these effector cells in the immune response. If you have an increased infiltration in this cell in the tumor, which is a very good sign, but it's not enough. Are these cells predominantly tumor-specific or bystanders, for example? Are they shared between the treated and the distant tumor? We need to understand what effect we have on the tumor vasculature, on the lymphatics, on the endothelial cells. Are we going to improve the trafficking of the dendritic cells to the lymph nodes, of the, for example, T cells into the tumor? One other point we did not really address is the innate immune response and its role in this overall antitumoral immune response. Its qualities are really important. The polarization of the myeloid cells, for example, or the macrophages M1, M2, that will determine whether this microenvironment will be more immunocompetent or pro-inflammation, pro-anti-tumor, or it will be more immunosuppressive, trying to turn down this inflammation and T cell, for example, effect on the cancer cells. Are we going to increase DC recruitment, DC activation? And finally, because we are trying to use a local therapy to generate a systemic effect, we need to understand what's going on into the lymph nodes, what's going on into the distant tumors. So several mechanisms have been postulated, and this is one example taken from a recent review paper discussing immunological effects of histotripsy for cancer treatment and trying to make sense of all very more and more abundant literature on preclinical models and to propose different mechanisms of action on how histotripsy will decrease pro-tumor immune cell, will increase the cellular immunity, and how all of this could result in an increased systemic immunity, especially when combined with immune checkpoint therapies. But there are still a lot of details and points that we don't really understand. And these are really important now if we want to think about designing clinical trials. And for example, what is going to be the most effective therapeutic ultrasound intervention? Is it to use it for drug delivery, for example, in the brain, or is it for ablation? If we think about ablation, thermal or mechanical ablation, and what fraction of the tumor should we ablate? What are the most promising approaches for combination treatment? What ultrasound modality will synergize the best with what drug? What the best timing for this combination? Should we start with immunotherapy or with therapeutic ultrasound or mixed combination? What patient tumor profile will benefit the most from this combination? We talked about a little bit about these neoantigens, for example. Is tumor mutational burden something that we should assess to try to profile the patient that we will treat? Will we treat primary tumor or metastatic tumors? And should we treat 1, 2, 3? What the best way to monitor the response of the tumors? What biomarkers should we assess? Can we monitor this response through peripheral and blood samples biomarkers, or do we need biopsies? And finally, what tools and approaches our community of therapeutic ultrasound will need in order to really move the field forwards in terms of new assays, for example, spatialomics, in terms of new bioinformatics resources, in terms of data sharing. And so to conclude, we know that HIFU ablation is a treatment option for oncological indications with a demonstrated efficacy of local tumor control. We have some, so far, mostly case reports that therapeutic ultrasound can elicit a systemic antitumoral response, but we need more proof to really be convinced that therapeutic ultrasound intervention alone will be sufficient to elicit this systemic antitumoral response. And most likely, what we will be going for are combination therapies, for example, with checkpoint inhibitor on TPD1 to really trigger this systemic response. And finally, one of the take-home message is that this modulation of the tumor microenvironment is a very complex interplay of factors, very complex interplay of sequences, and that we need further research to improve our biological understanding. And that will be really critical to optimize our treatment regimens and to optimize clinical trial designs. And I want to leave you with some resources. So associated with this MOOC is a list of review papers, of recent review papers. We released a public bibliography database with the FUS Foundation, listing the publication in the field of the first immuno-oncology. All these publications are annotated, so you can make very specific searches by treatment modalities, trypsis, thermal ablation, and so on, by tumor type, even by tumor model for preclinical studies. And we used all of this to review all the knowledge, and we share the landscape analysis that you can also find in the resources. And finally, we also published guidelines for immunological analysis to provide some guidance on what type of assay should we perform, what type of time points for the analysis we suggest, we recommend. This is available also through our website, and you find a link in the resources in this MOOC, and this should be published very soon. So thank you very much, and if you have any questions, don't hesitate to contact me.